So ELISA, for detection of antibodies in a patient sample, right, indirect, known as indirect by most people, right, or antibody capture. So that's what we're actually going to do today. ELISA is actually an acronym. It stands for Enzyme Linked Immunosorbent Assay, right, and that's because, as we've seen, right, the secondary antibody, the second antibody we'll use, has an enzyme linked to it. So the little Pac-Man represents the enzyme here, and the Y shape represents the antibody, in this case the secondary antibody. So what are antibodies? They're a product of your immune system, right, made by specialized white blood cells called plasma cells, which are derivatives of your B lymphocytes, commonly referred to just as your B cells. And antibodies bind to specific antigens. And what antigens are foreign substances. And typically our immune system will respond by producing a specific antibody against that antigen. So this is a specific interaction. And then also what we're using today is an immunoglobulin antibody. Immunoglobulin is actually another name for an antibody. But it's a little bit easier than saying anti-antibody antibody. That gets to be a bit of a tongue twister, right? But that is basically what it is. It's an antibody against an antibody, right? So that's our secondary antibody. Remember what I said it binds to, the primary antibody. It doesn't bind to the antigen, right? This is a special antibody. It binds to an antibody, right? So it's an anti-antibody, meaning it binds to an antibody. Antibody. I know, it's a tongue twister. It'll make sense as we go through it. All right, so this is the general structure of an antibody. And each of your B cells produces a unique antibody. And this is the general structure where you have two light chains, which are in dark here. And these are protein chains, two of them that are identical, and two heavy chains that are in white here. And they come together to form up here at the top what we call antigen binding sites at A and B. So this is where the antigens bind to the antibodies, okay? So this would be your antigen, right, and the antibody is specifically binding to it. And that's because each antibody is different in this portion, so it can bind to different antigens. So where then does the antibody bind to the primary antibody? Where does the secondary antibody bind? Well, we're creating a sandwich, we're creating layers. So this is the bottom of our plate, right? Then this primary antibody that's specific to the antigen, and then down here in this heavy chain portion, this part that sticks out, or what some people call the stem, is where the secondary antibody is going to bind. So why are these different colors? It's representing that each one of these antibodies is different, right? That's what this color representation is showing you. And as I said, right, each antibody up here in what we call the variable region is different. And it's going to bind to a different antigen. But for each antibody, both of these sides are the same, right? It's like a mirror image of itself. Okay. The rest of it, the little bit of the light chain and especially the stem portion of the heavy chains, these remain constant. So that the structure always is the same, right? We always get this characteristic Y shape when we're dealing with these. In addition, your immune system responds to 5 different heavy chain sections here that can be made. There's 5 different classes of antibodies. There's IgA, IgD, and Ig again stands for immunoglobulin, right? And the letters stand for the different classes. So you make different ones under different circumstances and your body responds to these differently. And we're not going to go into the detail of that in this course, but just know that there are different types of antibodies out there. So what specific portion of the antigen does the antibody bind? It's called the epitope. So it's a very molecular interaction between this antibody and a small piece of the antigen. So it doesn't grab onto the whole thing. It grabs onto a molecularly specific section. And that molecularly specific section on an antigen we refer to as an epitope. right? So different antibodies will even bind to different sections of an antigen, right? So samples that we typically can use for these types of tests is we can take your plasma. This is the liquid phase of your blood when it's not allowed to clot. So this is, they'll draw it with an anticoagulant in the tube. And then when they spin it out, they can separate out the cells, the red blood cells and the white blood cells, from the liquid component. In the liquid component will be your proteins, like your antibodies. But also the clotting proteins will be there as well because they were not utilized. Me as an immunologist, when I had to run tests and they would give me plasma, I was not happy because those clotting proteins can sometimes interfere with the action of this test. Better sample would be a serum sample. And this is when the blood is taken in a special tube and it is allowed to clot. So all the clotting proteins go in the clot and they don't end up in that liquid phase, and that liquid phase is referred to as serum, right? And typically that's what we prefer to use. You could use plasma, they'll be there, just you also have those clotting proteins that can get in the way, right? So serological testing, right? Typically we're talking about serum, right? Serum is the sample that we're utilizing. And again, it doesn't have those clotting factors. It'll have other things, right? The antigens, the antibodies, and other proteins that are found in the liquid phase of our blood, just not those clotting factors. And there's a lot of them. So they can really gunk things up. So what are we doing today? Antigen or antibody detection? We're going to do antibody detection. So we're going to be doing indirect versus direct ELISA, right? And I'll go to this at the end. On Blackboard I have a link to BioRad's animations, but I've taken all their pictures. I'll tell you, you just have to hit the little play button and it goes through each one of the pictures that I'm going to show you. There's no audio, so don't be freaking out, right, that you don't hear anything. And they have both there, both animations. So they have the direct, right, which remember, we're detecting for antigens in that case if we're doing a direct test from a patient's serum, and we're using a specific antibody supplied that will only bind to the antigen we're looking for in that sample. So here are some more specific examples, right? Rapid strep test, ovulation, pregnancy, drug test, HIV antigen if we're looking for the antigen, various flu tests, the rapid flu test for that virus. Now, indirect, which is what we're going to be doing today, where we're looking for antibodies, we're going to be using a purified antigen. So we know, right, what antigen we're starting with, and we're looking for if that person has the antibodies to that specific antigen, right? And so again, in that serum sample, there should be antibodies. So HIV antibodies can be detected from serum samples this way, and other samples, for a matter of fact. Heartworm can be detected, those heartworm antibodies, as well as the hepatitis viruses, whether you made antibodies against A, B, or C. This way they can detect for that. For both types of ELISA tests, you make, as we said, that antibody sandwich, right? We're going to start out with our antigen, right? This is your key to everything, right? Your antigen, your primary antibody, which, of course, what is it binding to? Just the antigen. Secondary antibody, what is it binding to? Only the primary antibody. This is important. And what does the secondary antibody have that helps us with this test? Has the enzyme linked to it, which is going to turn over our substrate and produce that color for us. This type of technology is used a lot in research, as well as we've already listed all the commercially available and in doctors' offices quick rapid tests. Sometimes the product can even be a fluorescent product, right, that it produces, or chemoluminescent products where they luminesce because of the chemicals. So, and I have in my office, I don't want to take it down off my wall, but Peter was kind enough to find a really cool picture. And so there are lots of labeled antibodies that are utilized with specific stain colors associated with them. So they're using primarily two here. They're using a red and a green. And then they're using a dye to stain the nucleus of the cells. And so they were able to stain specific molecules. And what this is helping is notice that, what do you notice about this? Look at the red, right? And then look at the green. What can you tell me? You can tell me location, right? So we don't only know it's there, we know where it is, right? Microscopically, they're looking at it on these cells and they're like, oh, look, this molecule is probably associated maybe with the membrane, it seems like, right? Where this is inside the cells, right? Who cares what the molecules are? I don't even have a clue. But again, just to show you, right, that even by this different coloring, and just like how we talked about with the fluorescence with the different sperm, right? How they made one green and one red, right? And here they're using antibodies to be able to tag, right? That's the difference here, is that there's these fluorescent dyes are stuck to the antibodies. So then the antibodies are sticking to the cells, and we can actually see where they're sticking microscopically. It's pretty amazing stuff.
Just so you know, this comes from the lab of Jerome Breslin, who is one of the guys I work for at LSU. He's my favorite ones.
Yeah. Because he produces cool stuff like this.
Yeah.
But yeah, I have a poster in my office, and it shows all different cells stained differently with different dyes, and they put it together in a rainbow. It's really beautiful. Science can be art too. It can be quite beautiful. So you're going to start out with a strip of labels, tubes, and you're going to label right on these little tiny microtiter tubes. And then you're going to add from a little tube some purified antigen. And again, it's just going to look like liquid you're sticking in. These are microscopic, you're not going to see it. But what's going to happen is as we let it sit, it's going to stick in the wells, right? And then we're going to wash, right? We're going to dump out the liquid, and we're going to remove any of them that do not bind. So, and we have to do this before we apply the primary antibody, right? So anything that's not sticking, we want washed out. The reasoning for this, right, and we're going to do it twice. We're going to rinse twice. Why do we wash? What did I say? We're making it stick, right? Is everything going to stick? No, right? If it doesn't stick, we don't want it there. We want it to be washed out, okay? So to remove any unbound reagents. Every time we wash, it's stuff that's not actually binding what it's supposed to bind to. If it's free in solution, it's not sticking, then it gets washed out. Because otherwise it's going to mess up our sandwich, right? So what happens if you wash too much, though? You could wash out all your stuff, right? And then you're going to end up with what type of results? Negative results. Those would be false negative, would they not? What if you don't wash enough? You're going to get antigens that are bound and antigens that are not bound in this case, right? And what's going to happen when you add the antibodies? They're going to bind to the ones that are bound and the ones that are not bound. The problem is the ones that aren't bound, what's going to happen to them when we wash? They'll get washed away. And also you have this competition now, right? Between the ones that are bound and not bound. We don't want that. We only want them sticking to the ones that are bound so that they will stay, right, in that tube. Make sense? Okay. So that's that in a whole lot of words. All right. So we're not doing the blocking that's indicated in your lab manual. The wash buffer that we use is significant enough to help the non-specific binding in the wells that may happen. So the first thing that you're going to do now is we're going to add positive controls, right? So you'll have tubes that contain what? What are we testing for? Antibodies. In this case, which antibody? Primary, the one that actually binds to the antigen, right? So in our positive controls, we've got to have the primary antibody, right? Otherwise, it's not going to come out positive. That binds to the specific antigen we're working with. So what's the purpose of positive controls? pregnancy test has a built-in positive control, right? 2 lines pregnant. One is the test sample, one's the positive control. If you don't get the positive control, the test will tell you, do it again, right? Inaccurate, right? Because if the positive control doesn't come out, can you trust the validity of the test? No, because if positive control doesn't come out positive like it's supposed to, something went wrong with that test, right? So the test system is simply not working properly. Something went wrong. What's the possible cause of negative results in all your positive controls? What could have went wrong here? Too much washing, right? You washed it all out. What else? Adding the wrong stuff to the tubes. And the reagents themselves could be expired. They only have a certain activity level. And again, if you're running a pregnancy test, right, and you don't get positive control, then it's not valid. So our positive controls are going to have the specific antibody to the specific antigen. So in this case, it's the black binding to the black. And then all the rest will be removed when we wash. Our negative samples, right, we're even going to do a negative control here, right, to be sure, because remember it's a multi-step process. What do you have to have in order for it to be positive that has to be left out of your negative control sample? The primary antibody. The primary, right? And this is so that we know that the secondary does not bind to the antigen, right? It only binds to the primary antibody. So if we get a positive result in these, this is not good, right? So this is to serve as a purpose, because this is a multi-step process, right? That you cannot get a positive result without that primary antibody. How could this happen? How could you have positive results in your negative controls? Failure to wash, right? Don't wash stuff out and stuff gets left behind. Contamination of reagents, cross-contamination when doing the procedure, which I'm going to teach you guys how to do it so you don't. Why would the positive control results and all the negative controls affect your interpretation of the results given by the patient sample? So if you get positive results in your negative thing, which you shouldn't, right? It invalidates this test, right? There's no way you could have reliable results, right? Something went wrong. Negative is supposed to be negative, right? So what's in our negative wells? It should just be antigen, right? No primary antibody binding to it. That's it. So the next thing you're going to do is actually run test samples. So you have 2 test samples numbered 3 and 4. One of them's positive and one of them's negative. You've got to find out today who's getting the good or bad news depending on what you pretend you're testing for, right? You can pretend whatever you want. So we're going to add those. We're going to do this in triplicate. Why are we going to do 3 wells for each one of our samples and for our positive and negative controls? More reproducibility, less likely you're going to make mistakes, right? More accuracy, more precision. You could even calculate standard deviation if we assigned a number to the color. Why would you get inconsistent results? Well, you guys are new at this, right? So it could be poor technique, right? Or poor sensitivity of the test. How good is it detecting at what's present? And of course, if you have extreme variability, this would cast doubt on your results for your patient samples. So, this is a qualitative. So this is positive or negative test that we're doing. But this test can be set up to be quantitative, where you can actually get a number. But we're not doing that today. So our patient samples that are positive, right, they'll have antigen with antibodies bound. Patients who are negative, right, we should just have antigen in the well, no antibodies, no primary antibodies. So again, we're going to remove, we're going to wash twice anything that's not binding, right, from all the samples. What would be the consequence of washing too vigorously? Again, we could wash out what's in there and end up with negative results. Get too many of our antibodies removed. We just want unbound ones. If you don't wash enough, again, we end up with this competition like we had before with the antigen. Now we've got primary antibodies floating around, and they could bind to our secondary antibody. And will really affect the sensitivity of our test. So we only want stuff that's bound. So at this step, we should have, for a positive, right, antigen with the specific primary antibody attached to it.
You know, I just added that comment that flew in, and that's, you really want to get rid of other serum components and all of the various antibodies.
Yeah.
Since your secondary antibody reacts to human. Right?
Yeah.
It
Right.
doesn't matter even if it's your target antibody.
Right. Yeah.
Is any antibody in there at all?
Good point. So the fact is, and what Peter's bringing to light, is the sample you usually are putting in is a bunch of different antibodies. And you really only want the one that's sticking to the antigen to stay behind. Any unbound ones, you need for them to be gone. Otherwise, it will mess up your test and give you inaccurate results. Because this secondary antibody will bind to any antibody. It's not specific, right? So negative, what should it look like? Again, just antigen, right? Just the specific antigen should be there. Then we're going to add the secondary. Remember, what is this antibody binding to? It's binding to the primary antibody. It's not binding to the antigen, right? It's binding to the antibody, helping us detect the presence of this antibody that is hopefully just stuck to the specific antigen we have here. Right? And then when we add our negative wells, of course, we won't have any of this. It'll just be good old antigen, right? Because there shouldn't be any primary antibody for the secondary to bind to. We've hopefully effectively washed out any non-binding. You're going to wash 3 times before you add your substrate, so that any secondary that's not binding to primary gets washed away. Because just the presence of that antibody, right, will cause the reaction. So again, if we wash too much, we're going to remove too much of the secondary, and we're going to end up with false negative results. If we don't wash enough, we're going to end up with false positive results, right, if that antibody stays behind. This can be a really crucial step. So again, how many times are we going to wash? The last time we wash? 3 times. 3 times. Then we're going to add our substrate, right? And I think I have some foil back there. This substrate is light sensitive, so I'm not going to give it to you guys until you're at that step, right? And what I'll probably do is, and then I'll give it to you, and you can add it and cover it up, and then peek underneath it. It literally only takes like not even 5 minutes, like a minute, and we'll get the reaction of the colorless substrate with the enzyme, and we'll get the blue color, right? And then, as I said, this can be done quantitatively if you were to titer out and knew the concentration of whether your antibody or your antigen, and read it on a spectrophotometer specially designed for reading the plates, the full 96 well plates, and you would be able to see it. So why is it important not to mix these things? Because specific things need to bind to specific things, right? And so when we wash, we're going to make sure we don't have overflow or cross-contamination as well. And so I'll show you guys that for the first step. And of course, we don't want to mix them in the tubes, because then that would cause them to bind beforehand, give us inaccurate. So these are some examples of different types of testing that's used in labs like Peter and I have worked in, where they're using membranes instead. And a lot of times too, they'll even separate out protein components and such, and with gel electrophoresis to put the different size, and then they're going to embed them on a membrane. And then again, the antibodies are applied with the different the enzymes or markers, so that we can see actually where they are, right? So we can actually visualize different bands on the plates. And then chromatography, right, where they're trapping stuff utilizing antibodies. So as I said, antibodies are used a ton nowadays for detecting and allowing us to even separate out things, be able to acquire a particular thing for an assay. So let me end that.